Thank you very much.
I should clap because of his summit.
Okay. Well, thank you. It's a great pleasure to be here, and I'm very excited to be in front of a group of individuals who are obviously very enthusiastic about what they do. And Rashid asked me to show some pictures, and I wasn't going to do this, but so I just, you've seen a few of them already, and it was just a little over a year ago that I was actually sitting on the summit of Mount Everest, with the emphasis of sitting. So it's a tough road to hoe to get there. But like all things that are difficult and challenging, you have to be well prepared, and you have to work hard. And so I can only say to you that doing difficult things like trying to summit Mount Everest is no more difficult than it is getting through a PhD or getting tenure or anything else. It just takes a slightly different set of skills and a little bit of different attention. Okay, well,
Paul,
I'm actually...
Paul, how many years did you spend to prepare for?
Okay, so my entire story now crashes because I had never climbed a mountain of any sort or done any of this 1 year before I summited. So I actually summited Mount Everest 359 days after summiting my very first mountain ever, which was slightly higher than Mount Washington. It was Mount Rainier, and it was 14,000 feet. And so I did set myself a task, a very serious task, and I learned how to do all of the things that are needed to equip myself to go into a rugged, horrible environment and survive. I won't say I thrived, but I think when I look back on it, it's an experience that it's difficult for a person who sits in an office reading papers and writing papers. You can't really compare those two things. Although I think getting papers published sometimes is more difficult than getting to the top of Mount Everest. I think most of us can understand that. Now I'm going to do some small battle here with my computer because for some reason it's not working well, but I'm just going to flick through these just to point out that you have to climb over 50 to 60 ladders through the Khumbu ice field. These are vertical ladders, cross huge crevasses. Some are 4 or 5 ladders across. That's actually me crossing that ladder. And you do a lot of practicing. This is a camp we made on the top of a 6,000 meter peak, just to practice camping on the top of a peak, where if you walked over out of your tent 5 meters, you would fall 5,000 or 6,000 feet. And so they made us do that, and that was a scary thing. This is our team. We had 28 climbers. This is actually a huge avalanche. It doesn't look very big, but it's about half a mile wide. So if you get the size of that, that went for about 4 to 5 miles, and that killed some people on our trek. So this happened frequently. This is about 7,500 feet just below where I'm standing, and it's 60 degrees straight down. So if you fall off that 1 foot wide track, you go at approximately 9.8 meters per second squared for about less than a minute, and then you stop. Yeah, eating food is interesting at that altitude. This is the yellow band on your right, the famous yellow band, which is between Camp 3 and Camp 4. This is just another ladder. It's quite steep in areas, climbing up sheer ice. This is going up to Camp 3, and you go to Camp 4 before you summit. It can be miserable. I won't tell you what's actually happening there, but it's pretty obvious that you get nausea and it's not very pleasant. This is a 5-ladder crevasse, This was on a practice. They sent us up to Camp 3 just to practice. Camp 3 is at 26,000 feet. So we went up there just to see if we could tolerate the environment.
Right.
This is a French colleague of mine. He fell off the ladder, but he was attached, fortunately.
But it's not just the practice, it's conditioning, right? It's,
Yes.
Your lung,
Yes.
it must be
It's making enough blood cells
Right.
so that your blood is like honey. It's thick. And you make many, many times the number of blood cells. Rashid, did you have a question?
Did everyone make it to the top, or how does
Um,
that work?
We had 28 climbers in our group. 5 of them dropped out of the team 2 to 3 weeks before we made the summit. We were there 9 weeks. It took 9 weeks to do this. And 1, 2, 3 climbers on the ascent did not make it. So 3 dropped out on the way up. 21 made it to the top of our team. This is just some of the equipment had to be taken up to Camp 3 and Camp 4. There's about 200 bottles of oxygen there. Just to give you some idea, it costs $685 to carry 1 bottle of oxygen to Camp 4. So that's what the actual cost is to get a bottle of oxygen to Camp 4, $685. And you need a few hundred of them. So this was at Camp 2 and Camp 3. Camp 3 is on the Lhotse face. The Lhotse face is one of the 14 8,000 meter peaks, which is next to Nuptse, which is next to Everest. They're 3 peaks together. And Lhotse is here, and our camp was here, right up on the face. Yes, this is a tent on Camp 3. You can see the slope which with the tents are. And it's about a mile down there. So you don't sleep much when you go there. This is coming down from the summit actually, and we got stopped by about 30 people because somebody couldn't abseil, or up above Hillary's Step, and somebody had some troubles, and we stopped. Yes, so this is a quick way to Tibet. It's about 10,000 feet down there to Tibet, and on the other side it's about 10,000 feet down to Nepal. So it's the 2 quick ways you can go. Quite a few people take those routes actually. Now this is my Sherpa, and we're sitting waiting on our way down from the summit because of some traffic below us. And you see there are several ropes here. There are many ropes. There are dozens and dozens and dozens of ropes on Everest. But there's only one rope that you must use for each year. So you pick by colour. And it's hard to believe, but sometimes you can't remember the colour. And you say, how can you not remember the colour? Because your brain is not working very well. This is Hillary's Step, and it's about 8,000 feet straight down. And you have to walk around this just nasty thing. It's best if you don't look. And this is near the summit, looking down. We're very, very high. Well, about 29,000 feet. More ladders. This is sitting on the summit, on the top of the world. And I was there for 40 minutes, and while I was there, a colleague behind me here had a serious episode, and one of my friends, and I was there for 40 minutes, and I had absolutely no idea that there was a problem behind me, sitting 3 feet behind me. And I found out because this is actually a video camera, and there's 2 or 3 of them here. This was filmed by Discovery Channel, and 3 Discovery Channel videographers went to the summit with us. And they were filming what was happening behind me, and I was sitting 3 feet away, and I didn't have a clue. I had no idea until I saw the videos when a few months ago, that one of my friends had a serious episode on the summit, and they were dealing with that. So it's interesting how you have no perception whatsoever of what is happening. So you could easily turn around and go the wrong way, or forget to come down. You remember to come down because the guides and Sherpas come along and prod you and say, come on, leave. You say, why? So because we have to go down. Oh, okay. So you lose all sense of reality, and it's scary. It's very, very scary. Rarely do you put yourself in a situation where you have no control over your mind.
So out of the 21 people who made it to the summit, how many of them made down to the
all of us made it up and down. Yeah, our team survived quite well. And if you want to know what the height in feet is on the summit, this is my climbing companion, who was my tent companion. He always climbed in twos. And I took a picture of his watch. It's 29,030 feet. So he was standing probably 3 or 4 feet off the summit. I don't know whether that would make any difference to the altimeter, but that's what it was. And you can't see this very well, but I have lost all the skin on my face because I got severely burnt. There was no sun. In fact, the sun was, we were in clouds. I didn't see a darn thing from the top of Everest. So all that time and effort, and I got up there and was just completely covered in clouds. But I took my mask, an oxygen mask, and eye mask off, and I suffered tremendous burns on my face, lost all the skin on my face. So anyway, all's well that ended well, and we got to the bottom, and we drank 32 bottles of champagne, 5 cases of beer, and about 4 bottles of whiskey. And about 2 days later, we recovered, and then we left. So, well, that's got nothing to do with the topic of today's seminar, which is about advances in cytomics, and isn't anywhere near as exciting as climbing to the top of Everest, but it's just as challenging. And what I'm going to talk about is some of the challenges and how we might address those challenges, and what new ideas might there be. Has everything that could be invented in the field been invented, as has been stated 100 years or so ago by rather famous scientists? And of course the answer is, well, no. So I'm just going to run over a few things and talk about a little bit of history, because I think it's relevant in a technological audience to see how changes in technology impact how discovery is made. And we can go back to 2600 BC in China, where lots of dyes were being developed and a great understanding of the chemistry of dyes. I think it's rather intriguing that many of the dyes that have been used for thousands of years are very similar to the sorts of dyes we find very useful in science today. And I just put that up there so you can get some perspective of how far back we go in terms of the technology that we use. Sometimes biology uses very old technologies because they're very appropriate, and sometimes we try to move on with more modern things. And I also think that it's rather interesting that if you look at the fundamental discoveries that we use, almost none of them are this side of the 1950s. I know that seems odd to you, because you think that everything was discovered the last few years, like the iPhone and the laptop and the MP3 player, and it's true that those are crucial discoveries for a younger generation. But most of the tools that we use come from these discoveries in the first half of the 20th century. And what we have found ourselves doing is extending those technologies, innovating on many of them, rather than inventing them. If I look at the field of cytometry, or cell analysis, measurement of cells, we would go, I would say, into the late 1800s, and people like Ehrlich, who really started to look at the history of stains, and he realized that by using stains that they could identify types of cells. Now we do exactly the same thing today, but we use more sophisticated tools like antibodies and enzymes and fluorescent proteins, for example. So we have a few more sophisticated tools, but actually we're just extending the types of tools that Ehrlich was using. And then there were obviously some fundamental discoveries, such as understanding the role of DNA and the functional nature of how cells were controlled by people like Casperson and Foilgen. And then I'm going to talk a little bit about a couple of discoveries, two in particular. One by a gentleman by the name of Papanicolaou, who some of you may have heard of, some have maybe not, and another, Wallace Coulter, and the subsequent discovery of cell analysis technologies by Mac Fulwiler. And I'm going to talk a little bit about where these tools go and what we can do with them. And I started this by thinking about what are the really key technologies. And I think probes are really important. And I started with that slide from 2600 BC. And we are still developing fluorescent probes. And there are now tens of thousands of different molecules that are out there. And of course, fluorescence proteins. Detectors are changing almost daily. But, interestingly, we still use the PMT as our basic detector, or the diode. And they haven't really changed that much, and certainly in our implementation. And perhaps those are areas where we might see some excitement one of these days. And of course, we can compute things now. And if I show you some early work, and you will see why that early work stopped where it stopped. Because in many cases, you couldn't do any computation. And it made it very, very difficult. Well, you could, but there's a limit to how far you can go with a slide rule. So without the modern computation tools, this really slows us down. And the other thing that computation tools does is it brings disparate groups together. So when you bring these sorts of tools into a mix, now you have informatics people working with biologists and engineers, and you'll see the sorts of things that happen. And you may not appreciate, some of you, younger people, how important the internet is. It's there, and it's always been there, right? So as far as many young people are concerned, email has existed forever, and probably in 5 years' time, Twitter will have existed forever. So what we see, those older people see, is how much change that has caused in how we do science. And so that means that many of us have had to change what we do and how we do it. But I think those are really important things in how technology has changed. So to just give you a couple of examples in fluorescent probes. So in the last 25 years, the books on fluorescent probes have gone from nothing to this thick. And that means that you can create, you can custom build a probe to suit almost anything you do. So chemistry is pretty important there. And I'm going to talk about the complexity of fluorescent probes and how that actually makes it difficult for biologists to make some measurements, and how we have to think about these in some slightly different ways. And then I talked about PMTs. This is an early PMT. And those PMTs were developed in the Second World War and weren't available to us scientists till some years after the Second World War, early 50s. And in the last 10 years, another revolution in terms of really quite sensitive multi-array PMTs. And this 32-channel PMT I've been using now for 10 years. And this is a wonderful device. Quite expensive, but allows us as biologists slash biomedical engineers to create some new types of measurements. And I've already mentioned the transformation in technologies. So if we look at cells and cell analysis and how that has impacted us, if we look at the early days, all you could do was look at cells under a microscope and stain them with stains that would change the color of cells. Even though the fluorescent microscope was probably developed around 1907, probably by Kohler, it wasn't well used because there weren't good light sources. And that caused a real problem. So there was a failure in technology availability. So colouring of cells became important. In the 1950s, engineers started looking at medical problems by saying, well, why do you count cells by hand? Because if you look at them under a microscope and try to count them, you'll get high errors because you can't count very many cells. So your coefficients of variation are going to be very high. How can you give accurate counts? And so we had a revolution in counting. And then in the 60s, as I'll show you, there was a revolution in how you can manipulate cells. You can actually move cells, grab cells, and do things with cells. That happened in the middle of the 1960s. And in the 70s was the invention of monoclonal antibodies. And that was a Nobel Prize winning feat by Kohler and Milstein in 1975. So until 1975, we did not have the ability to manipulate biology with such precision as identifying receptors and markers on cells and tagging just those cells. We had to use broader tools of making polyclonal antibodies to very broad spectrum receptors. So that was transformational in the 1970s. And then in the 1980s, when we had the cell counting tools, we had the cell sorting tools, and we had the ability to identify them, immunology took off, and one very important thing transformed the field of cell analysis and cell biology in general, and that was AIDS. In 1980, it was discovered that there was a syndrome that altered the balance of white blood cells in such a way that caused the immune system to completely fail and resulted in certain death. And these discoveries were made because we had already in the last 7 to 8 years discovered what this white blood cell population was composed of. When I was an undergraduate student at university, I did my first class in immunology. And in that class, we learned about 2 types of lymphocytes: large lymphocytes and small lymphocytes. And I remember having a scientific debate in our class. It was divided up into 2. It was only a small class of about 10 people. And half the class had to argue that small lymphocytes were the most important lymphocytes, and the other half argued that large lymphocytes. That sounds funny, doesn't it, today, when we know there are many, many types of lymphocytes, but we only know them because of 2 things. One, we learned how to separate cells with the cell sorter, and 2, we learned to identify cells with great precision with monoclonal antibodies. So you see how one thing has led to the next, and we see this iterative process here of discovery, many times invention, and I'm going to show you where this will lead to innovation. Okay, well, I've spent a lot of time on that slide, probably more than I should have, but let's look at what the pathway for normal discovery is. We have a problem, we have a hypothesis, we have an experimental approach. What we do is we look for the tools that we have available to us, and we use those tools in the best way we can. But what happens when we can't reach a solution? Well, what we do is we create a new tool. And my argument here is that the only way to do that is integration of technologies, is multidisciplinary way. And so when we do that, and we bring biology and engineering together, we end up with a new technology and we end up with a solution. I'm going to show you two examples of how this transformed cell biology. And first of all, I'm going to point out to you a few fundamental discoveries that were needed along the way. In 1953, the sheath flow principle that allowed us to place cells in liquid and track them and know where they were. And that was Crosland-Taylor's invention in 1953. Of course, you can't get past at the same time the fundamental understanding of the nature of nucleic acid. Same year, in fact. Same year, the invention of the confocal microscope. Now, it wasn't built, although Minsky did design such an instrument. It's thought that he might have tried to manufacture one, but there was no evidence that he built it, but he designed it in that year. And in that same year, Wallace Coulter commercialized the cell counter. And from that cell counter, we had a transformation of technologies. Now, that cell counter actually caused a problem. So here's the story of how an invention is made. Wallace Coulter built this device because as an engineer, he observed that the physicians in the hospital couldn't get accurate blood cell counts. And in the 40s and 50s, the easiest test to do was a blood cell count. Now, you can tell a lot of information if you know what the blood cell count is in somebody. You can tell if the red cells are normal numbers or the white cells are normal numbers and what type of cells there are. Because we knew in the 40s and 50s that there were lymphocytes and monocytes and granulocytes. We knew that they could be divided into eosinophils and basophils and neutrophils. All this was known. We knew what platelets were. Now, we didn't understand the nature of each of these cell types, but physicians knew that if you had too many red cells, or too few white cells, or too many monocytes, or too few lymphocytes, that related to a clinical syndrome that was well defined. Wallace Coulter said, I can build a device that will count those cells accurately every time. And so he did build that. Now, what happened was that that device then went out into research labs. And one such research lab was Los Alamos National Laboratories. And this is in the 1950s and 1960s. Now, a pathologist got hold of one of these devices, and some engineers at Coulter said, well, not only can we measure the number of particles that are in this sample, but we could put a pulse height analyzer on there, some new technologies, and we could look at the nature of the particles in there. So they did. Well, they didn't explain to everybody how these things worked. So a pathologist got hold of one of these devices. So what he did was he measured red blood cells with this pulse height analyzer. And what he discovered was, to his surprise, was that there were two populations of red blood cells. Now, this was inconsistent with the medical knowledge of the day. So it would seem like it's an incredibly exciting discovery. When that happens, it's really crucial for colleagues to review our data. That's what we call peer review. And this is where group meetings and discussions amongst our colleagues are very important, because sometimes the discovery that we make might be artifactual. It might be that we're missing something that's maybe obvious, maybe not so obvious. It turns out that there were some engineers who looked at this and said, it doesn't make any sense from an engineering perspective that there's two peaks of red blood cells. It doesn't, it goes against the medical knowledge. And the pathologist said, no, I've got two populations of red blood cells. So what happened? Well, what happened was that this gentleman here, his name is Matt Fulwiler, was an electrical engineer. He decided that the only way to prove that the pathologist was wrong, that this bimodal population of cells that existed, was to separate those two populations of cells. And he thought to himself, how could you do that? Now, this is where the other part of dissemination of knowledge occurs. He had been to a meeting and heard a talk by another gentleman. And that gentleman's name was Richard Sweet. Now, Richard Sweet had a different problem. Richard Sweet worked for the Air Force. And his problem was he wanted to measure high-frequency variations, and he needed to map them. And you could not get a pen on a paper and a plotter. This is before digital technologies, guys. So we had pens and paper and plotters in those days. And he couldn't do it fast enough at, 100 kilohertz or faster, 200 kilohertz. And he said to himself, there's got to be a better way. How could you get ink onto paper faster than by a pen? Well, you know the answer. How do you do it? Oh, you don't use inkjet technologies anymore. I forgot. That's old technology. Well, he invented the inkjet printer. Mac Fulwiler had been to a talk, and he had seen the fact that he was firing droplets at a piece of paper, and he said to himself, hold on, if you could fire droplets at a piece of paper, couldn't you put cells into those droplets and fire them somewhere and then catch them? If you can move a droplet around, you could probably do that. And this is what happened. Isn't it amazing that in the old days we had paper and people wrote letters? This is a letter written in 1964, and this letter was written by Matt Fulwiler to Richard Sweet, and he said, I find your direct writing oscilloscope to be a clever and fascinating device, and I greatly appreciate receiving whatever detailed information you provide. Now, Twitter wasn't there then, but 5 days later, in the 1960s, when there were horse-drawn vehicles, he received a reply in the letter from Richard Sweet saying, wow, this is a very interesting idea. After giving your ingenious cell sorting scheme additional thought, my only conclusion is that it will surely work. This is real discovery taking place. These tools didn't exist. So, here's the rub. 8 months later, 8 months, not 5 years in a PhD, 8 months later, Matt Fulwiler designed and built the very first cell sorter. And this is a unit which we have in my institute, which was built in 1967 by Matt Fulwiler. It's the second one ever built, and it's the only one that still exists. This thing actually worked. And he was able to separate those two populations of cells that you see here. And guess what he found? Answers? Come on. What did he find? Okay, there's only two possibilities. One is that there's two populations of red blood cells that are different, and the other is that there's not. Who's going to vote for there's no difference between these red blood cells? 1, 2, 3, 3.5. Any advance on 3.5? I highly encourage advances on 3.5. There was no difference. This experiment was done in one afternoon. One afternoon. They spent 8 months inventing a new technology to look at red blood cells. And what did they find out? Nothing. There wasn't even a paper in it. Dear science, we made a discovery. There's no difference in red blood cells. Doesn't go down, does it? That afternoon, I spoke with Mac Fulwiler one day, and I said to him, Mac, what did you do? He said, well, I went up to that same pathologist, and I said to him, is there anything else in blood other than red blood cells? And he said, yeah, there's white blood cells. And he said, okay, can we sort white blood cells? So there you have the history and the beginning of how the cell sorter was invented. And it was in Marvin Van Diller's lab. This is Mac Fulwiler, who did the invention. And of course, it was Richard Sweet's inkjet printer. And the collaboration, Richard Sweet sent Mac Fulwiler one of his precious devices for shooting ink onto paper so that he could try it. This is sharing. This is scientific interaction and collaboration. Well, tools have changed in the last 40 years, and we have all sorts of technologies that are out there, and so we can do amazing things with these. But I'm going to give you a second example of another invention. Yes?
But before you go on, red blood cells are known to be special. There's no nucleus in the cell. So is there something that, for example, when you do these things using inkjet printer type of devices, you kill the cells very often?
Well, that's interesting. They actually worked out how to do this, and everything had to be done in saline solution, because if you're going to sort, you have to be able to put a charge on this. And so they're able to protect the cells. In fact, the very fact that they were able to do this in 1965 amazes me, because I'm not sure that we could be that clever today and be able to make such inventions so quickly and make them work. But of course, the way the cells were going through the orifice was the same orifice that he put on here they had on the Coulter calendar. They were actually measuring impedance. And so they really weren't measuring two populations of cells. They were really changing the nature of how the cells were going through the orifice. So it was an artifact. Let me give you a second example of an invention that wasn't meant to happen. And this is related to Papanicolaou. And this is related to the fact that for 65 years, 70 years, we've been trying to work out how to diagnose cancer. And it's a tough one. And the original diagnosis of cervical cancer was developed by a scientist, a pathologist by the name of Papanicolaou. And what's interesting about this is that every pathologist, every single pathologist that today diagnoses cervical cancer was trained by Papanicolaou. How can that be? Papanicolaou trained a bunch of people, who trained a bunch of people, who trained a bunch of people, who trained a bunch of people. So what you have here is technology that is in the brain. It's a neural network called the brain. And that's half the problem, because what we like to do is convert that into technologies that actually could do this in an objective way without having to be trained necessarily and have the only information go from one person to another. If any of you have ever been in a pathology department of a major hospital, you'll see pathologists sitting at a long table with a big tube in the middle, one microscope, and about 6 sets of oculars. And they're all looking at the same thing, because they're all training their minds to understand, when they look at an image of a piece of tissue, to understand, is it normal or is it abnormal? And it seems to be a fairly simple decision-making process. Turns out it's incredibly complicated. This gentleman actually worked for IBM. And this photo is a photo taken of Lou Kamensky in about 1960. And he worked at Watson Labs at IBM. And he is the inventor of character recognition technology. Has all the patents in that. It was invented by IBM. Now the reason they were doing this is rather intriguing, and I won't go into it. But it's got to do with the CIA and the Russians. And if you can work it out, then good. But it's all about secrets. Anyway, that's the technology that was in his lab. And he was working on this. Now, another pathologist comes into this, and it was his next door neighbour. And one day over the back fence, they were talking. What do you do? Well, I look at things under a microscope, and I try to work out what is cancer. Oh, what do you do? Well, I try to work out what a J is, or what an I is, using mathematics and computers. What are they? Well, they're things that do computation. Huh. So you could pick an I from looking at a page. Yes. Okay, good. Why can't you pick cancer cells from non-cancer cells? Very quick conversation this was. And he got intrigued by that. And so he said, well, let's try that. And he spent, actually he's retired now, but he spent the last 45 years trying to do this. And it's a very, very difficult thing to do. But in the end, he built a microfluidic thing. You might look at this and think it was just in lab tech engineering last week, or microfluidics journal last month. Actually, that was published in 1963. Microfluidics is just a little older than the last few years. And he was actually able to sort cells in 1965 using microfluidic devices and capture them and do things to them. Exciting, eh? So, this is an instrument that he built in 1965. It had 4 sensors. It measured 4 angles, 4 fluorescent channels, measured light scatter. It had a computer in it. Actually, it had most of the computers that IBM owned at the time. And they built 3 or 4 of these instruments. Huge technologies. This is built by IBM in 1965. So IBM obviously made a quick decision soon after that big computers would be more profitable than big medical devices. Probably made a very smart decision. But this was the technology that was being built in the 1960s. So, several questions now arise. What can we do today, or what should we do? Has everything been done that could be done? What could we possibly do? How could we advance the cause? What are the new opportunities? And at the end of the day, one of the things we're interested in is always finding that elusive cell. When we find that elusive cell, what do we do with it? Can we manipulate it? So those are the questions. And you know, there's now about 20 companies, one right here in this town, that build cell sorters. And this is really advanced technologies. And there's probably 100 companies, maybe more than that, maybe 200. There's probably 100 in China alone making fluorescent probes. And then lots of people making these sorts of tools. And then analytical software. All of these things need analysis. So the current state of the art is the following. You can collect tens of millions of cells. And you can collect them and purify them. You can do 18 simultaneous channels. So that if you look under a microscope, you can see 3 colors, right? Red, green, and blue. Pretty much. That's it. But on a flow cytometer, you can do 18 colors simultaneously. But it's a little bit complicated. You can run an exceeding 100,000 cells per second and sort them. And they've moved mostly to the digital domain. And of course, there's fast ways of doing things. We do these things for stem cell transplants. Even in clinical laboratories now doing 10 colors in routine. And so it is a quantitative technology. And so cytometry, as its name implies, is a measurement tool. It's measuring cells. It's understanding cells. So the real issue here is you can't do these things without bringing diverse tools together. So, I asked this question, has everything been invented that isn't? And I'm going to talk to you about 2 things that are sort of transformational for the field. And some of these have been around for some time. And the first one I'm going to talk about, we finished this work about 6 years ago. So it's still not commercialized, although one company has taken a license on it, and a second company is looking at it, and it'll be 5 more years before that makes its way into a commercial machine and probably works well. So it takes a long time to get some tools into technology. And then I'm going to talk about high throughput, because at the end of the day, sometimes doing things quickly is important. Cells don't live forever. And if you want to do a lot of assays, you might have to do it quickly, because you can't keep those cells around or they change. And so I'm going to talk a little bit about where we're going with high throughput, because it's opening up the area of drug discovery. And I'm just going to refer, excuse me for referring to a Purdue faculty member, but David Glengarry is really the father of hyperspectral imaging. He's an emeritus professor at the university and was one of Professor Bashir's mentors when he was a student at that university. Very famous person, and he of course developed the technology that we use in Landsat and hyperspectral tools. And I won't go through that because you all know what they do and how they work. But in cell biology, we use these tools. And we're trying to use these tools to allow ourselves to separate features of cells and understand them better. And very simple tools. This is a slide that I wouldn't normally show to an engineering group, but biologists like simple explanations sometimes for complicated things. And they've all been to the paint shop and got a paint chart. And what I do is I show them how this looks like if you were in a flow cytometry lab and you converted this into a dot plot. And this looks like a cell sorter dot plot. And of course, these are all gateable populations. So if you can do this with a paint chart, maybe you can do this with microbeads or maybe with cells. And so the idea here is to really expand our ability to find probes and see probes. I already told you there's thousands of fluorescent probes, but you can't use thousands at a time. And if you do use 18 probes at a time, you need 4 different lasers. You need them separated spatially. And you've got to do a lot of complicated stuff to make it work. And so the question is, what can you do with moving into the hyperspectral domain? Now, a normal flow cytometer looks like this. It has a laser and it has a delivery system which sends liquid which has cells in suspension. In flow cytometry, everything has to be in suspension. You can't look at tissue. So you put the cells into suspension and you shoot them past the laser beam. And as they go past, you measure properties. The scatter from the laser is one, and then the fluorescence properties are others. And that's what it looks like. And the way, what we do is we have all these probes. We work primarily in the visible domain in biology. And there's a number of reasons for that. One of them is that we like to be able to see what we're doing. And the tools were first invented for the visible domain because the only things we had were microscopes. And so we followed that pathway, although there's lots of new opportunities. But you see the problem here. These are the tools that we want to separate, and they're very difficult to separate. This is how we do it. You get a PMT, and you put a bandpass filter in front of it, and you say, I'm going to collect this band of light. It might be 20, 30 nanometers. And you get another one, and you get another one. And you expand this as many as you can. And this might come from one laser, and you might get 3 from another laser, and 3 from another laser, etc. Okay? So this is the process that you work. And then you have a couple more parameters of side scatter and forward scatter, and you now have a sort of multivariate data set which you can work with. And we call this polychromatic cytometry. If you move to hyperspectral cytometry, you've got to actually try to collect a spectrum, a whole spectrum. Well, that's very difficult because you don't have enough photons. You've only got about 10 to the minus 5 seconds to make your measurement, and in that 10 to the minus 5 seconds might sound like a lifetime to a fluorescent molecule, but it's a very short amount of time to collect photons and in cells. And so this is a 32-channel PMT that I showed you. And what we basically do is, as cells go past, we build up information in histograms, and we calculate how many cells fit whatever parameters or variables that we measure. If we move to multispectral cytometry, we change that paradigm, and instead of collecting individual bands, what we do is we say, look, that's a fingerprint of the cell. And remember, we're looking at a single cell, just one cell. Not looking at a suspension of all the cells. We're looking at one cell at a time, and that's it. So now we have sets of data that actually give us a fingerprint, and that fingerprint might look like that, or it might look like that. And so you ought to be able to pull that fingerprint out. Now, it's not easy, because you've only got 32 points. So the resolution is not great. But it turns out using moderately simple techniques such as principal component analysis, you can separate, and if you go iterate through this, you can pull out each of the colors of these populations by going through iterative processes and end up separating dyes. And here's an example of how you could actually get something of use. So here are 3 dyes. They turn out to be chlorophyll, rhodamine, and phycoerythrin. 2 of those dyes are very commonly used in mammalian systems, and one is obviously related to plant systems. But if you put those 3 dyes together, you couldn't separate them. They are in the same band. Here they are here. They're totally on top of one another. There's nothing you could do would separate them. But if you actually collect them, just using a 32 channel PMT, and you apply principal component analysis without anything else, without any gating, nothing, you can separate these out absolutely perfectly. And now you have ways of pulling overlapping probes out. And so that really opens up opportunities that we in cell biology didn't have before. And then you can play with your data till your heart is happy, and do whatever you wish. So I won't go into these data because time is almost up. How long do I have for this talk? 3 more hours, okay. Sorry. So you can see that it gets a little bit complicated, but it's more fun because there's a great challenge here. As how do we extract all this information? So much information now. And now the fun is for a different group of individuals to pull that out. That's what makes this a lot of fun. So I want to talk just a few minutes about another area, and I'm going to just finish this section by pointing out to you a what-if scenario. So this is a picture that I've used for about 7 or 8 years. And as far as I'm aware, it still can't be made. But it could be made. Someone will make it one day. And it's a mythical probe, a nanocrystal, that apparently could be barcoded in many, many ways. We sort of went overboard when we sort of, in my first slide that I ever used of this, I think I had millions of combinations on it. Well, that was sort of outrageous. It's possible, I suppose. But if you could make these tools, we can measure them. That's the point. And we could measure them in cells or on cells or around cells. And so the idea is to innovate by integrating technologies so that when somebody else comes up with this invention, that it will be integratable into our field. And I just throw that up there because that's sort of a holy grail of where we might go. Okay, so I'm going to jump to the last piece of this presentation. I'm going to talk about automation and how you make things go fast and how you analyze things quicker. And I'm going to use as an example of this the problem that pharmaceutical companies have, and that is drug discovery. And in drug discovery, one of the things we need to do is understand the properties of drugs and how they affect cells. Because you can have the best drug in the world for killing methicillin-resistant staphylococcus, but it also kills all your lymphocytes, and you die. So you've succeeded in killing the infection, but you've killed the patient. So it's really important to know that what the toxicity or what the other roles are of the molecules that are made. And these days, it's being done in cell systems. And it's quicker in cell systems. And it's more useful sometimes, particularly if you're using human cell lines. So this is a driving force for technology development. And one of the problems is we can't go fast enough. We can't get the information quick enough. Now, I published this paper about 20 years ago. And the interesting thing about this paper is it's hardly ever been referenced, which is usually not something to brag about. But it's been referenced about 4 times. I think 3 of them was me. But it turns out that this was an interesting invention, and nobody cared. And the reason nobody cared, because, well, you couldn't do anything with it. And about 7 years ago, what the idea was, by the way, and I wrote grants to every possible institute that you've ever heard of, and then some. And everybody said, this is a really stupid idea. And so don't get too upset about getting your ideas trashed, because one day they might come back to haunt you. Anyway, the idea was we wanted to do automated diagnosis. Well, if you have all this information about blood, why can't you put it back together quickly and extract the real information from it? And what we were able to show 20 years ago, we could identify acute lymphoblastic leukemia, myeloid leukemia, ALL, CLL, chronic lymphocytic leukemia, all of these syndromes, simply by looking at the patterns of expression of the lymphocytes. And it seemed to be too good to be true, because these were the days of backpropagation neural networks, which were the latest things. And people sort of said, well, you can't really do that with simplistic approaches to data analysis. And we came up with this idea and didn't go anywhere. So I'll leave it at that. But a few years ago, another fellow by the name of Larry Sklar in New Mexico reinvented that technology. And what he did was he just put it on a robot and he made it go fast. Now, just to show you that one of the problems in cell biology is that there's so much information that you can extract out of cells that you don't know what to do with it, and you can't even put it together. Here's an example from a fellow by the name of Gary Nolan at UCSF. And what Gary Nolan is doing is he's taking lymphocytes, he's staining populations of lymphocytes, these are white blood cells, he's staining them with 7 different colored dyes. They're actually the same dye, but 7 different intensities. And then he's separating out those populations, staining them with 10 different antibodies each, then mixing them back together and putting them through a flow cytometer and getting an enormous amount of information on hundreds of thousands, if not millions of cells from humans, and trying to do clinical diagnosis. And he's succeeding in doing this. It's an amazing feat. So that's being done using dyes and antibodies. So this is probably going to lead to very significant automated diagnostics. So, going back to Larry Sklar, by putting robots on systems, he was able to speed up the system. And so we took the same technology and then did very similar things to Sklar. And our goal was, how do you take very high numbers of cells, millions of cells, and hundreds of drugs, and screen them very, very quickly? So I can tell you that we can screen something like 32 drugs with 10 concentrations of each drug And it takes us 12 minutes to run all of those millions of cells, and in that 12 minutes is not only the collection of the data, but the reduction of those data to, I see 50 curves, or actually any type of curve you want. And what that's allowed us to do now is to do experiments that you would never be able to do before, because in just a few minutes you can do amazing studies on cells. So you can do time-lapse studies where the time is very short, 5 or 10 minutes, when you could only do a couple of samples before in 5 minutes, maybe 5 samples in 5 minutes. We can now easily do 200 samples in 5 minutes. And we probably can double that without any trouble. So this now opens up possibilities of really, really advanced types of studies that couldn't be done before. If you integrate, for example, the hyperspectral technology with the high-speed technology, then you have some really fantastic possibilities. And I think it'll be some years before those sorts of things are integrated, but the ideas are there and it is possible to do them. So let me summarize a very long presentation, and I apologize for how long this has been. But we did have to climb Mount Everest first, and so that's, I'll use that as a good excuse. So at the end of the day, I'm going to say that innovation is just as important, if not more important, than invention when it's coming to developing new technologies. I think that we have a long way to go with fluorescence both in maybe in the Raman domain. We haven't really done very much in live cell systems. All sorts of opportunities there. In fluorescence, there's been a huge amount of work, and I think there's a great opportunity there. I think that now that we can do things fast and furious, we'll ask questions we couldn't ask before. And think about if you could parallel these in microfluidic systems, just like Kamensky built in 1965. So if he could do it in 1965, you might be able to do the same thing today, I suspect, especially with the tools that we now have for mass production of these things. I will say you can't do any of this, and you shouldn't even try it, if you don't have somebody in your group who understands informatics, because it's a pointless exercise. It's managing the data that becomes the really, really big challenge in cell biology today. And that's an area that I think is pretty exciting. So I sort of put a bit of, on the bottom there, sort of new probes and new computation, new technologies. When you add that to things like high content and high throughput, that is getting more information out of our cells and running them through faster, it opens up a new biology. And that new biology means, to me, new diagnostic and faster and cheaper tools, and for you, new opportunities that I think you can all go down. And so let me conclude with acknowledging the people in our group that have done most of this work, and the collaborators that we have, some colleagues from here at Eyesight in this town, and the funding bodies that have funded us and the companies that have supported us. Thank you very much for your attention.